Hi, in this video we're going to talk about two things. We're going to talk about independent assortment and dihybrid crosses. But first, let's talk about or remind you of something that we talked about. We talked about the principle of segregation. Now, the principle of segregation we defined as, I'm going to give you just a second to think about this. What did we define principle of segregation as? Hopefully you said it's the separation of alleles during gamete formation. Now, we've talked about this. So the key here is remembering it's the separation of alleles. The reason why that's so important is because we're going to talk about the principle of independent assortment. An independent assortment is the same exact definition as principle of segregation with one word difference. So again, the principle of independent assortment is the separation of blank during gamete formation. And it's the separation of genes. So let's demonstrate this. Let's draw this. So if we have, let me draw these a little bigger. So if we're going to draw a homologous pair, and one homologous pair codes for a red structure, we have another, the other homolog codes for color of that structure as well, but it's going to code for a blue on whatever structure we're talking about. During segregation, the code for red, the allele for red and the allele for blue will separate. That's the principle of segregation. The principle of independent assortment goes on and talks about genes. So if we have a second, So if we have a second set, put that back in there, second set of homologous pairs, and if we've got this one that codes for long, this one would also code for long. The reason why sister chromatids both code for the same thing is they're an exact copy of each other. So if these two code for long, we're going to say this one codes for short, therefore this one would code for short. Now, if we go through meiosis with all four of these chromosomes, with these homologous pairs, at the end of meiosis, we are going to have 4 daughter cells. Each of those daughter cells will have 2 single chromosomes in each cell. We're going to have 2 that contain chromosomes that code for a red structure, 2 that contain chromosomes that code for a blue structure. I need to draw in the rest of my chromosomes. We're going to have 2 that code for, if this were to split down the middle, just like this, if we get a red chromosome or a chromosome that codes for a red color, we're also going to get the sister chromatid that becomes the single chromosome that says long. So again, when we have the red in this situation, we get long. And on this side with the blue, we get short. Now, the only reason we got short when we got blue or long when we got red was because of the way these homologous pairs lined up. So let's look at a second situation with the same homologous pair, only this time this side is where the short lined up and the long lined up over here, and the colors lined up the same way they had lined up previously. And you can see when this cell divides right down the middle, just like so, we're going to end up again with 4 daughter cells. Draw that one a little bit bigger. We're going to end up with 4 daughter cells, and those daughter cells are again each going to have 2 single chromosomes. 2 of them are the ones, we'll have 2 of them that say short. Oops. 2 of them will say long. And 2 of them will be red. So are the ones that are red going to join or going to be in the cells that contain short, or the ones that contain long? So you can see with this dividing down the middle, and then during meiosis 2, this is going to divide here, this is going to divide here. Our red are going to be with our short. So we're going to have a chromosome that codes for red with this short, and a chromosome that codes for red for this short. And the blue is going to be with the chromosome that codes for long and the chromosome that codes for long. So as you can see, depending on how they line up, how the homologous pairs line up, we can either get red with long or we can get red with short. And that all depends during meiosis how those how those homologous pairs line up with each other. This is called the principle of independent assortment, because we have a separation of genes, and this separation of genes gives us these variations. Now, we can actually we can actually have a violation of the principle of independent assortment. So let's draw this again. Draw that a little longer. There we go. That should be drawn a little better. Let's do this again. Okay. So again, we've got long, we've got short, and we've got red, and we've got blue. But let's pick another genetic trait. Let's say we've got right here on this chromosome, right there, it says 5 digits. In other words, like 5 fingers, 5 toes. So this one codes for 5 digits. This one is also going to code for 5 digits. Well, this one over here and this one over here, these code for 6 digits. Okay, so when we go through meiosis 1 and meiosis 2, we're going to divide here and we're going to divide this and we're going to divide this. We again end up with 4 daughter cells. We again end up with 2 single chromosomes in each of these cells. We again end up with long. Well, let's do the colors first. We end up with red and red, and we end up with blue and blue. And we end up with, when we have red, we have long. So on this one, it's going to be long. And this one's going to be long. And this one's going to be short. And this one's going to be short. Now, on the red chromosome, we've got 5 digits. We've got 5 digits. I'm going to just put a D there. And this one I've got 6D for 6 digits. And this one I've got 6D. Now, question. Can one inherit red and 6 digits and long? Is this possible? Well, as you can see here, anytime you're going to inherit red, you're always going to inherit 5 digits. So the answer to this would be no. The reason why is because you can't inherit red and 6 digits together. The reason why is because they're on 2 separate chromosomes. This would violate the principle of independent assortment because the genes cannot separate from each other. They're actually stuck on the same chromosomes. So this is the principle of independent assortment. So if you would like to take a picture of this screen, please do so now, because in just a minute I'm going to clear the screen so we can talk about a dihybrid cross. All right, we're going to talk about dihybrid cross. Okay, so we're going to talk about a pea plant. We're going to cross 2 pea plants, as a matter of fact. We're going to cross a tall pea plant that has purple flowers. We're going to cross that with an additional tall pea plant with purple flowers. Now, tall is dominant to short. So these two are the same gene. They're 2 alleles for the same gene. And purple flowers are dominant to white in pea plants. So we need to clarify this individual or pea plant is heterozygous for both traits, and this one is heterozygous for both traits as well. So this is the phenotype of this generation. We're going to call this generation the P generation. So what we want to do is we want to cross these 2 plants, and we want to find out what we get for our F1 generation. Well, in order to do this, we need to know what the genotype of each of these plants are. So if they're heterozygous, that means we're going to have an uppercase and a lowercase of each letter. So in the first case, we're talking about tall, these plants being tall, we're talking about height. So over here, we're going to say that I'm going to use a capital T for tall, and I'm going to use a lowercase t for short. So this individual is heterozygous for height. So I'm going to put a capital T and a lowercase t. They're also heterozygous for purple flowers. Purple is dominant to white. Now, when I write these 2 P's, it's very difficult to tell which one is capital and which one is lowercase, or if either one of them are different. So what I do with my lowercase p's is I always put a line underneath where it would be as though it were sitting on the line. It clarifies that the P is lowercase versus my uppercase P's. So a purple heterozygous flower would be capital P, lowercase p. And this individual would be the same thing, capital T, lowercase t, because it's heterozygous for height, and it is heterozygous for color, so it's demonstrating that dominant color. Once we have the genotype of the excuse me, once we have the genotype of the individuals, we then need gametes. In order to reproduce, we need gametes. Now, when you're trying to figure out what the gametes are, if you remember FOIL stands for first, outer, inner, last. So we're going to create 4 gametes, just like we talked about earlier in this video. So we need the first of each of these 2 genes. So our first letters are capitalized, capital T, capital P. So that's first. Our outer letters are the ones on the outsides of these. So we have a capital T on the outside and a lowercase p on the outside. The I stands for inside, so that's the lowercase t and a capital P, because those 2 are the 2 letters on the inside. And our last are the last of each letter. So we have a lowercase t and a lowercase p. Now, this individual is the same genotype, so it's going to produce the same gametes. First, outer, inner, and last. Now, once we have our gametes, this determines the size of our Punnett square. Since we have 4 types of gametes here and we have 4 types of gametes here, we're going to have a very large Punnett square. So we're going to have a 4 by 4 Punnett square. I apologize for my angles here. It looks kind of weird, but you get the idea. So we're going to create a 4 by 4 Punnett square. And we're going to take the gametes from one individual and we're going to put those gametes across the top. Anytime the 2 gametes are circled, or excuse me, anytime the 2 letters are circled, that means they're within the same cell. That's one gamete. So if they are circled, they need to go together on one column. So if they're within the same circle, they need to go together on the same column. Again, capital T, excuse me, lowercase t, capital P are together, so they go together on top of the same column. And the last one, lowercase t, lowercase p, are together in the same cell, so they go in the same column. Now we're going to take these 4 gametes and we're going to write these 4 gametes down the side. Again, capital T and capital P are in the same cell, so they have to remain together. Capital T and lowercase p are in the same cell, so they have to remain together. Lowercase t and capital P are again together, and lowercase t, lowercase p are together. Now we're going to fill this in just like a multiplication table. So we have a capital T and a capital T. We have a capital P and a capital P. We have a capital T, capital T. We have a lowercase p and a capital P. So we always put the capital letter of the pair first. So we have a lowercase t and an uppercase t. So the uppercase t is going to go first, and 2 capital P's. So we have an uppercase t and a lowercase t, and we have an uppercase p and a lowercase p. Do we have 2 uppercase t's? We have an uppercase p and a lowercase. And continue filling these in until you have all of your boxes filled in. Takes a few minutes to get these all filled in. but it's important that you're careful that you don't make a mistake. Now, these are, let me scoot me over there. So these are all the possible genotypes of the offspring. So we're not going to talk about ratios of the genotypes. That would be crazy numbers. But let's talk about phenotypes. So the possible phenotypes of this pair, this individual and this individual, if we cross them, what are our F1 genotypes? So let's look here. We've got F1 genotypes. So what combinations of these could we possibly have? So we have tall, short, purple, white. What combinations could we have? We could have tall, purple. We could have tall, white. We could have short, purple. And we could have short, white. All right, so let's go back over here to our Punnett square. We have, let's see how many of each of these types we've got. So let's take a look here. We see here that this is a tall purple. So let's put a tally mark here. We see this one is also tall purple. And this one is tall purple. This one is, wow, tall purple. So that entire top row. This one is also tall purple. Then we've got this one. This one is not tall purple. This one is tall, but this is tall white. This one is another tall purple. This one is tall white. This one is tall purple. This one is tall purple. This one is, white. Or excuse me, short purple. This one is short purple. This one is tall purple. It's a lot of those. This one is tall white. This one is short purple. This one is, finally, we have one that is short white. Now, anytime we take two individuals that are heterozygous for two traits, and cross them, we will always get a 9 to 3 to 3 to 1 ratio. We always get 9 dominant, dominant. So we've got 2 dominant characteristics. 3 dominant, recessive. So dominant trait, recessive trait. Recessive, oops. Dominant and recessive, recessive. So we always get this ratio in that order. Anytime we have a dihybrid cross, anytime we do heterozygous, heterozygous crossed with a heterozygous, heterozygous, we get a 9 to 3 to 3 to 1 ratio. We get a 9 to 3 to 3 to 1 phenotypic ratio. Now, that's the biggest Punnett square you'll ever do. So if you'd like to, take a picture of this screen so you have it to save, or you've got your notes. So let's go ahead and clear this. And let's take different offspring. Let's see, let's pick, I don't know, let's go short. Let's go short purple. Let's go, I don't know, let's go tall white. And let's see what kind of Punnett square we have for this. So again, this is our phenotype. We need our genotype in order to get our gametes. If we talk, and we're going to say this is heterozygous for purple, and we're going to say the tall is heterozygous. Now, we don't have to say whether the tall or the short is heterozygous or homozygous, because since it's short, short is recessive. So tall is dominant, short is recessive. So I have a capital T and a lowercase t. So anytime we have short for pea plants, we know we have a lowercase t, lowercase t. Purple is dominant to white, so we have a capital P and a lowercase p, because purple is dominant to white. And again, we're using that P, so we need to make sure we underline our lowercase. So with tall, if we're heterozygous, we have a capital T and a lowercase t. And for white, white can only be found with two lowercase p's. So let's talk about our gametes. Now, we said we would FOIL. So our first is capital T, excuse me, lowercase t, capital P. Our outer is lowercase t, lowercase p. Our inner is lowercase t, capital P. And our last is lowercase t, lowercase p. But you can see in this situation, we have these two that are exactly the same. So we can ignore the second one because they're the same. We have these two that are exactly the same. So we can ignore the second one. So we can see that this individual only produces two types of gametes. So let's take a look over here. If we FOIL, we have first of each letter, outer of each letter, inner of each letter, and last of each letter. Then when we examine these, we see that these two are the same, so we only need to keep one of them. We see these two are the same, so we don't need to keep the repeat. The nice thing about doing it this way is you'll notice there are two gametes here and there are two gametes here. The wonderful thing about this is we can do a 2 by 2 Punnett square instead of a 4 by 4, which is great. Saves a whole lot of time. So again, we keep our letters that are in the circles together. And this is why it's so important, because with this P not being underlined, you can tell it's a capital P, even though it's very small. Should be a capital P. So we have a capital T, lowercase p for this one. We have a lowercase t, lowercase p for this one. So we have a capital T, lowercase t, capital P, lowercase p when we combine these two. We have a capital T, lowercase t, and two lowercase p's when we combine these two. We have two lowercase t's, a capital P, and a lowercase p. We have two lowercase t's, two lowercase p's. Now, talking about our combinations, we can have tall, tall purple. We can have tall white. We can possibly have short purple or short white. Now, When we look at this, we need to look and see how many of each we've got. So tall purple, this one is tall purple. This one is tall white. This one is short purple. And this one is short white. So in this situation, we have a 1 to 1 to 1 to 1 for our phenotypic ratio. Oops, ratio. There we go. Now, if I ask you this, what is the probability of having a short plant that has white flowers? And the answer is, since we're talking about probability this time, it's either 1 out of 4 short white, this is the only one out of the 4, or 25%. Okay? All right, so now that we've gone over this, now you have enough information to work on the handout, the dihybrid cross practice. And if you have any questions, let me know.